Have a good day, ladies and gentlemen. Today, I'm going to present the CRISPR gene editing, how we can use this technology to cure and repair the genetic disorders. So to do that, actually, we have started a competition that lasts for 4 years. And in that competition, Rare Disease Challenge, RaDiChal competition, serves CRISPR gene editing technology to cure rarely seen genetic disorders. So to do that, especially we need to go back for 20 years. So the first genome sequencing projects, when it is published, we have learned that almost 100% of all human genome is determined. However, this news are not yet completed, because in the couple of years later, we understood that 8% of human genome was not characterized correctly. To do that, we need to wait 20 years. And in 2022, a science magazine literature has been published to say that we are ready to sequence 100% of human genome. At that time, we were ready to understand the genetic disorders and all the mutations that can place in the human genome, as we understood the all genomic sequences in a single human cell. With the help of this genetic background and breakthrough, we now can determine the mutations and rare disease-related genetic disorders. So to be able to read was easy. However, to cure and to repair the mutations is the other challenging point in the genetic manipulations. So in 2011, Jennifer Doudna and Emmanuel Charpentier published a paper especially focusing on genome editing. And with the help of this technology, we can use a single guide RNA and a CRISPR-associated enzymes to be able to target any site of human genome. So with help of this, we are ready to manipulate genetic disorders to cure and repair the mutations, at most 89% of all genetic disorders. So with the help of this technology, there is more than 5,000 different genetic disorders that has the mutations in the specific sites, and the studies reported that almost 89% of these mutations can be repaired by genome editing using CRISPR approaches. Why CRISPR is better until now, especially because of its speed and price. Simplicity and accuracy help us to design quickly any genetic modification approach to cure genetic disorders. So with the help of this discovery and breakthrough, in just 10 years, now we have thousands of different literature showing the mutations can be fixed by CRISPR gene editing. Especially in just 10 years, there are a bunch of different CRISPR tools have been developed, and with the help of these technologies, not only the editing, but also the other modifications like insertions, like mutation repairing, and the other targeting and editing approaches help us to increase the efficiency and efficacy of the CRISPR gene editing. So when we come up all human-related diseases, not only the genetic disorders, but also inflammations like viral infection diseases or bacterial infectious diseases, we can use the CRISPR-Cas systems to target viral and bacterial genomes in the human body to degrade all the unicellular pathogens. On the other hand, we can use CRISPR gene editing technology to cure the cancer cells by manipulating oncogenic genes and the mutations on that kind of genes. However, when we look at the genetic disorders that cause rare diseases, especially because it is rarely seen in one person in 2,000 people. We can use CRISPR gene editing like modifications or modify by the CRISPR-Cas systems. Now we can perform gene knockout and gene replacement to cure several of these diseases. At the same time, base editing and prime editing technologies using CRISPR-Cas baseline, we can now modify any single base related genetic disorders that can be seen in cardiovascular, brain and neurogenetic disorders, lung and ocular diseases, and etc. So you can see that bioindustry of human agriculture and animal-based productions has been revolutionized by the CRISPR gene editing. Especially when we focus on human health, we can see that there is hundreds of different diseases and that is caused by monogenic disorders. For example, Huntington, muscular dystrophies, beta thalassemia, and other diseases have been shown that CRISPR-Cas system can target these monogenic mutations and cause repairing for fixing the disorders. So in 2019, we started to see that this kind of genetic modification techniques can be used in real human clinical trials and in ocular diseases and other heart-related diseases. Now, there is more than 100 different clinical trials have been started to cure the rarely seen genetic disease, starting from beta thalassemia, hemophilia, and several other rare cancer disease. So to do that, especially to be able to educate the students and researchers, we have started a competition called Rare Disease Challenge. In that competition, RaDiChal competition, we focus on thousands of disease, as we know that just 5% of this kind of rare disease have the medicines available. However, 95% of disease have no any therapy approaches, and that's why gene therapies are the future therapy, especially CRISPR-Cas modification system. So that now more than 3,000 different clinical genetic treatment studies have been started, and especially several percent of them are using the CRISPR gene modification. That's why by using CRISPR-Cas systems, we can aim to modify 75,000 mutations that can cause genetic disorders. So when we come up to all rare diseases, we see that at most 400 million of rare disease patients have been found in Earth. That's why using CRISPR gene editing as a future therapy would be the breakthrough, especially for genetic disorders and rare diseases. To do that, especially each year, starting from 2020, teams are prepared from different universities and just apply to radical.com. And when come up, you can see that there is some detailed file, and you can understand how you can design a gene therapy project. And especially you need to learn, first of all, what is CRISPR-Cas system and how we can use this theoretical approach in the experimental stage to cure genetic disorders. And after you register your university team, you are ready to go in a one rare disease gene therapy. To do that, first of all, we select three different rare diseases each year, and one university team should determine their target disease, and then they come up with 5 different researchers to build a team and try to get training and design a project. And of course, in the rare RaDiChal competition, teams getting together to perform several social awareness events, especially to tell genetic therapies, CRISPR gene editing modifications to the public, so that the world try to learn that new CRISPR-based therapy approaches can cure the rare diseases. It has been unsolved for long years. Until now, in 3 years, using radical competition from 6 different countries, at most 400 students from 89 different universities come together to cure 9 different diseases we have determined in 3 years. And to do that, especially the students, first of all, need to learn how we can manipulate the gene and do the gene therapy. To do that, we have filmed 12-hour, 4-lecture gene therapy recordings in Udemy, and by participating in this competition, students try to take theoretical approach for CRISPR-Cas gene editing training. And with the help of these theoretical trainings, the teams getting ready to design their gene therapy. Until now, we have focused on 9 different genetic disorders, and with the help of these university teams, using 90% of CRISPR gene editing approaches, now we have at most 90 different projects using CRISPR to target these diseases, such as multiple sclerosis, glycogen storage disease, Friedreich ataxia, and etc. So when we look at in detail, starting from 2020 to 2022, we can see that for each target diseases, there are at least 10 to the several different genetic therapy projects using CRISPR to cure this disease. That's why CRISPR is the future also for rare disease and genetic disorders. So this is from the final day from last year, and there was tons of different university teams, and they did their presentation and poster presentations by giving the information about their gene therapy projects using CRISPR approaches. And when they are evaluated by the judge, they're getting some points from their genetic therapy project, and the champion is the highest point collected gene therapy project. And when the university team getting champion, then they get the right to participate our laboratory, and we are gonna give the CRISPR-Cas materials to them to start their project, to start their in vitro experiments. So until now, we have published that this global inter-universities rare disease gene therapy project competition especially is the future for training and educating the researchers how to use the CRISPR-Cas systems in rare disease and especially genetic disorders. So as we know that there are at most 5,000 different rare diseases, so that we have a very long way to cure, to understand many of the disease. At least we know that hundreds of millions of patients are waiting this kind of future therapies. This is the first example from our competition, and our champions for CRISPR gene editing using team designed a prime editing technology. Prime editing is the developed based on CRISPR-Cas system using the reverse transcriptase that is connected to Cas nickase system. And with the help of this system, you can just modify single nucleotide up to 40 base pairs of deletions in a target space. So the first team, after getting their championship, they designed prime editing technology to cure spinal muscular atrophy. To do that, especially they designed the guide RNAs, and these guide RNAs are also having the mutated sequences to cure this after they produce the lentiviruses, they just target a certain spinal muscular atrophy cell model in laboratory. They just modify these cells using prime editing technology. To understand how prime editing technology affects the cell, they then just evaluated survival motor neuron protein expression using flow cytometry approaches. And this is the first results from the RaDiChal competition. And the first champions, they proved that prime editing specifically modifies single mutations on survival motor neuron gene. And they show in just 1 month, you can upregulate the SMN protein expression after modification using prime editing. So this is the first results, especially using the prime editing and CRISPR-Cas systems in rare genetic disorders. And this is the first clue that if we can target the right space, you can modify and repair the mutations that might be the future for many rare disorders. And at the same time, we have started the new organization, CRISPR Circle. Especially, we are aiming to perform several presentations and theoretical and experimental education to researchers to understand all circle of CRISPR gene editing technology. So this is our scientists, they are giving the lectures on new Cas enzymes, artificial intelligence to use CRISPR-Cas systems, CRISPR modified stem cells, and the CRISPR and ethics. Especially ethics are also important to be able to think how CRISPR gene editing technology can be used in human genetic modifications. And at the same time, we have given several theoretical lectures. And in that 3 hours, the researchers got the theoretical education about what is CRISPR first. And then after the all educational lectures, they will understand the theoretical how they can design their CRISPR gene editing technology to cure or modify any targeted space. This is the kind of pictures from our laboratory in CRISPR Circle. More than hundreds of different researchers got the education from me, and then we show that how we can use CRISPR-Cas systems in bacteria and the human cells. And with that CRISPR Circle organization, you can have the experience, theory, and idea at the end, and you will learn that how we can manipulate any genetic disorders with the help of these organizations. So I want to stop with Jennifer Doudna's words. So CRISPR isn't just a tool, especially for genetic engineering. Especially, it is a very powerful weapon for understanding how life works. Thanks for listening to me, and whenever you want, please do not hesitate to contact me for your question. Thank you.